Welcome to all viewers and well-wishers of the world. Purpose of my lecture is to learn the knowledge from the world, return the knowledge to the world. Motto of my life is learn, earn and return. As I have learned a lot, earned a lot, now it is the time to return a lot to the world. The topic of today's discussion is basic bacteriology. Bacteriology, as by definition, is the branch of science which deals with the study of the bacteria. True bacteria are those which infect the human being. So these are in one animal kingdom that is called eubacteria. Eubacteria are also common in the environment. Its second group of the environmental bacteria are in the second kingdom, which is called archaebacteria. But these are not going to infect the human being. So these are morphologically similar, but they have the major molecular and biochemical differences. Now, introduction of the bacteriology is that bacteria are the plural of the bacterium, a unicellular microorganism cause diseases in the humans and the animals. These are most abundant in the earth. They are in trillions and trillions. They can live independently. They are prokaryotes, means without any nucleus or nuclear membrane. So these are called prokaryote as opposed to the eukaryote, which are with nucleus. Genetic material is in the form of the spiral nucleotide, as in the eukaryote it is the nucleus. Cell contents are closed in a cell membrane. Cell membrane is surrounded by the cell wall, which is composed of the lipids and the proteins. This is a peptidoglycan complex cell wall, which is unique in the bacteria, and the gram-positive bacteria do have this peptidoglycan about 20 to 80 nanometer in thickness, while in case of the gram-negative bacteria, it is 5 to 10 nanometer in thickness. They are slight thinner than the peptidoglycan in gram-positive bacteria. Here one can see the peptidoglycan cell wall which is surrounding the bacteria and the contents of the bacteria which are inside. These are the ribosomes, the plasmids and the nuclear material in the form of DNA. Here again one can see the bacterial cell anatomy, the pilus, the fiber-like, hair-like structure by which the bacteria can swim or get attached to the other surfaces or the host cells, ribosomes, capsule which is surrounding the cell wall, and the nuclear material which is found in the center of the bacterial cell, and the cell membrane which is surrounding the cytoplasm. So the bacteria contain ribonucleic acid, which is called RNA, and the deoxyribonucleic acid, which is the DNA. Ribosomes are present in the cytoplasm. Flagellum is a long whip-like structure in cell wall which helps in the locomotion, or one can say these are the legs of the bacteria. Some bacteria contain hair-like structure called fimbriae or the pili, which help in the locomotion and attachment to the host cell, and it also help in the swimming of the cell. Plasmids are the packet of the mobile genetic information transferred from one bacteria to another. While the bacteria undergo the conjugation, many bacteria have the external capsule or the slime layer also. Bacteria multiply by the different ways, which is called the cell division. It can be the binary fission, it can be the conjugation, or it can be the sporulation by spore formation. Here again one can see the chromosome, the ribosome, the different part, the capsule or the slime layer, and the cell wall which is being surrounded by the slime layer and the capsule, and the other structure and the long tail-like structure which is called flagellum, which is the legs of the bacteria. Majority are the pathogens, they are classified according to the staining, shape, metabolism, and reproduction. They can be aerobic, which likes the oxygen, or anaerobic, do not like the oxygen. They can be motile or non-motile, maybe saprophytic, living on the dead organisms, dead material, parasitic, living organism, or symbiont. They can help the host in which they are living. They have capacity for the rapid growth and the reproduction. They have got the remarkable capacity to adapt to the changing environment by mutation, so they can live in the host for a longer time and comfortably. Can be photosynthetic, using light, or chemosynthetic, using the inorganic chemicals as source of their energy to live. Most are heterotrophic, absorbing nutrient from the environment. And one should know what is the difference between prokaryotes and eukaryotes. As I have already told you, all living organisms on the earth are composed of one of these two types of the cells. The difference is the cellular structure organization. There are three fundamental characteristics which distinguish prokaryotes from the eukaryotes. As you know, the eukaryotes do contain the nucleus and the prokaryotes do not contain the nucleus. So the prokaryotes are the smaller size. They have got the absence of the complex membrane bound organelle in cytoplasm, and they also have no nucleus plus the nuclear membrane. So these are called prokaryotes. Now coming to the difference between the bacteria and the human cell. Bacteria, as you know, these are the prokaryotes, and the human cells are the eukaryotes. Going to the size of the bacteria, these are 0.5 to 2 micron meter, and the human cells are usually more than 5 micron meter. Peptidoglycan in the cell wall, which is unique, it is present in the bacteria and absent in the human cell. Microtubules, these are absent in the bacteria, but these are present in the human cells. Ribosomes, though these are present, but these are of the different types. In case of the bacteria, these are 70S type, and in case of the human cells, these are 80S types of ribosomes. Flagella, as you have seen already, present in the bacteria and absent in the human cells. Teichoic acid, these are present in the bacteria and absent in human cells. LPS, that is the lipopolysaccharide membrane, which is seen mostly in the gram-negative bacteria. These are present in the bacteria and not present in human cell. And the other differences, one can see the nucleus and the nucleolus. These are absent in bacteria but present in human cells, as the nuclear membrane absent in bacteria and present in human cells. Golgi apparatus, that is absent and that is present in the human cells. Mitochondria, these are also present in the human cells but not in bacteria. Plasmid, these are present in the bacteria but absent in the human cells. And endospores, these are only the quality of the bacteria to make it. So these are present in the bacteria and these are absent in human cells. So here one can see the difference between the prokaryotes and eukaryotes. So one should not be disguised by the prokaryote and eukaryote. One have to be sure that whether this is hen or this is rabbit. There are certain things which are seen in hen and certain in the rabbits. Now, how you are going to name the microorganism? The Linnaeus established the system of the scientific nomenclature. Each organism has got two names. One is the genus and other is the specific epithet. Scientific names are the Staphylococcus aureus. It describes the clustered arrangement of the cells. Staphylo means clusters, and the golden color of the colonies, that is the aureus. So that's why it is called golden and aureus. Similarly, Escherichia coli, it honors the discoverer Theodore Escherick and describes the bacterium habit, large intestine or the colon. So Escherichia coli, the name of the discoverer plus the habitat. Scientific names may are abbreviated with the first letter of the genus Staphylococcus aureus, and Escherichia coli are usually written as S. aureus and E. coli. E. coli. Here one can see that Theodor Escherick, the German Austrian pediatrician who discovered this Escherichia coli, and the name is E. coli because of the name of Escherick. Now the shape of the bacteria. This is dependent on the rigid cell wall. Whatever the shape of the cell wall, that is going to be the shape of the bacteria used in the identification of the bacteria. According to the shapes, microscopic appearance, bacteria are divided into different shapes. These are cocci, singular coccus or round. This is bead shaped. Coccus means berry, as you have seen the berry. This is usually round shape. That is the Streptococcus and Pneumococcus. These are all cocci and the round in shape. They measure 0.5 to 1 micrometer in diameter. The other shape is the bacilli. Singular is bacillus. These are rod shape. Bacillus means rod, so these are rod shape. Straight cylindrical cells. These are of E. coli, Salmonella and Proteus. Mostly these are the gram-negative bacteria which are of this rod shape. In some bacilli, the length of the cells is equal to the width. So these are different from cocci and bacilli, but collectively they have got some shape of the cocci and some shape of the bacilli. So these are called coccobacilli, and these are different from the bacilli because of their size and shape. So one can see the cocci, the berry shape and the rod shape. So these are the cocci and these are the bacilli. Similarly, the other shapes of the bacteria are the vibrios. The singular is vibrio. These are curved or common shape, comma-shaped rods, and their name is because of their vibratory movement. They are comma-shaped and have a different movement from the bacilli. These are the vibratory movement. It means they are motile with a single flagellum at one end, and they show a rapid darting motility as in case of the Vibrio cholera. So they just shoot like a gunshot from one place to the other. The other shape is spiralia. Singular is spiralium. These are regularly coiled, rigid spiral or the helical forms. These are motile with the groups of the flagella at both ends. So these are seen in case of the spiralium minus. Here one can see the shape of the bacteria. They have got the flagella at one end. So that is why they have got the motility because of the presence of these flagella. And here one can also see the flagella at one end, and these are one can see the comma shape and the spiralium shape bacteria. The other are spirochetes. Singular is spirochete. Name is derived from the spira, means coil, and gete meaning hair. So one can see if the spiral hair of any person coiled. So these are the spirochetes. These are loose spiral forms, tightly coiled, motile organism by the rapid body movement, not by the flagella. Examples are the Treponema pallidum and Borrelia. Then there is another shape which is called actinomycetes. These are the branching filamentous bacteria. Their morphology resembles to the filaments, fungi, and sometimes these are considered to be the fungi and not bacteria, but they consist of the prokaryotic cells, not the eukaryotic cells, as the cells of the fungi are the eukaryotic cells. Some actinomycetes resemble molds by forming externally asexual spores for the reproduction. So these are considered to be the fungus, but these are basically the prokaryotes and not the eukaryote. Here you can see the tight spiralium. These are the coils, and that is the shape of the bacteria according to these coils. And here one can see the branching filaments, and that looks like to be the fungus on appearance. The other bacteria which are having some different shape, these are the wall-less or the mycoplasma. They lack cell wall, and these are bound by only the single triple layered membrane. They do not have stable morphology but occur as the round or the oval bodies. These are usually considered to be the smallest size of the bacteria, and these are also called the wall-less. Then pleomorphic, it means many shape, the different shape or the variable shape, like Coccobacilli. Several species of the bacteria are able to change their form, especially after being grown on the artificial media. And these organisms which show variation in shape or form are pleomorphic, and many shape do not have the fixed shape like cocci or the bacilli. Then there is a variety which is called rare form. These are the protoplast, spheroplast, and L form. These are basically formed due to the defective or the absent cell wall during development, or when these kind of bacteria are exposed to the antibiotics or the high salt concentration. They change their shape to these rare form, and these are called protoplast and spheroplast. As one can see, the gram-positive bacteria, under certain pressure, they can change to protoplast, and similarly, gram-negative bacteria can change to the spheroplast, and they have lost their cell wall and the cell membrane that becomes present. Similarly, the L form, one can see the walls of the bacteria. When these walls are lost due to aneurysm, these are called the L forms of bacteria. Then the shapes of the bacteria, these are just the summary of that which I have told you already. These are the pleomorphic, different shapes, filaments like the actinomyces, star-shaped bacteria, boss-shaped bacteria, appendaged bacteria, and sheathed bacteria. These are the different bacteria which get their shape according to the environment or where they are being grown, so they get their shape. So the summary of the bacteria is, as you can see, that is the berry-shaped cocci, diplococci, when two berries are combined together, when they are in clusters, the berries in cluster, these are the staphylococcus. When they are in chains, the berries, these are the streptococcus. And here one can see the bacilli, the spirochetes, as you can see the coiled tight hairs of any person, so that gives the spirochete shape, and spiralium just like the spring with the very tight coils, so that is called